Hello again. My name is Conor Gomes. I'm the lead developer of the SCISSOR instrument here at Pion. I'm here to talk to you today about some impressive science that was published out of the Institut Max van Laue alongside the University of Manchester, the University of Strathclyde, and AstraZeneca regarding probing how the microscopic and macroscopic contributions in diffusion impact a drug's delivery mechanism. Specifically, the authors look at small drugs and protein biologics in a supramolecular peptide hydrogel to probe these properties. The backbone of a supramolecular gel involves a small molecule, and in this case, the authors employ a FMOC-protected diphenylalanine, which under specific pHs spontaneously gels where these rings pi-pi stack, forming space in between each molecule where aqueous or other constituents can freely flow, but the interactions between these rings make it so that it forms a sort of a pseudo-solid structure where there's slow degradation over time or non-fluid-like behavior like lack of flow among inversion, as can see in B. And the APIs of interest that were impregnated within this gel can be seen below, the first being ibuprofen, to be compared to a previously study that the authors carried out. And the biological molecules are insulin and a lysozyme. These molecules were present within the bulk solution as the gelling occurred, and therefore after the gelling took place, these molecules were then present within the overall gel structure. After the formation of those gels, the physical and chemical characteristics of each were probed using quasi-electron neutron scattering by the time-of-flight indirect geometry spectrometer IRIS at the ISIS facility in the Rutherford Appleton lab in the UK. This method will probe fast dynamics and try to get physical characteristics and diffuse characteristics on very fast timescales in the nanoscale region. And it may be specifically used for probing solution dynamics because they will move and have some momentum on that time scale where the skeletal structure of the gel may not be able to be probed using these methods. In addition, within the quasi-electron neutron scattering, some backscattering and time of flight or BATS option in the ion 16B spectrometer at the Institut Laue Langevin. This was implemented to probe slightly longer timescales and measure the center of mass diffusion of things actually moving further and further through solution. To those smaller timescales on the neutron spectroscopy part of the study, neutron spin echo was also implemented, where a signal much like that of NMR, where a radiation is pulsed and you watch on longer timescales the polarization of a particle go back to its original state, and then the occurring echo signal that comes from that. To these microscale studies and very short time scales, much longer time scales in the hour region and macro scale with bulk drug release studies were done within Pion SCISSOR instrument, the subcutaneous injection site simulator, where these gels were loaded into our ECM cartridge, and then the drug release in each of these forms was then tracked via in situ analysis. To quickly review the results that were found from the IRIS spectrometer. So that's the smallest time scales. That would be measuring solvent sort of motion through the skeletal framework of the gel. So you can see here diffusion coefficients and residence times that are dependent on fitting a jump diffusion model, which would imply that the water is trapped at some lattice spot within this pseudo-solid gel structure, and then at some time point it's able to move and jump to the next free, what would be considered like a lattice hole. So at different temps, we can see that the diffusion coefficients for each of the solution and when that solution is confined within the gel, the diffusion coefficients increase with temp, as would be expected from a faster flowing solution, and the residence times of each of those waters decreases as if each of the waters is flowing faster. That means the residence time in each of those holes is decreasing. The BATS data was fit to Lorentzian curves to deconvolute sort of where the motion was coming from and what type of motion they were detecting. So based on the energy, what energy was being interacted with, and how much of that energy was being transferred, the authors could tell whether the signal was coming from a freely flowing particle like a protein in bulk solution versus a protein that was closer to the gel matrix that was sort of adhered by it. And so, based on all fitting of all these Lorentzian curves, the authors were able to fit diffusion models to the ibuprofen, the insulin, and the lysosome solution and gel formulations. With the ibuprofen, a jump diffusion model was fit appropriately, most likely because these small molecules will move through this matrix in sort of a jumping fashion like the water. With the insulin and lysosome, Ficking diffusion, where they are simply adhered based on their size through the matrix, and interactions with the overall skeletal structure is what's causing them to either gain or lose diffuse characteristics. And so, however, one interesting thing is that the insulin seems to move through the gel slower than the lysosome did. And that is probably due to the insulin's tendency to aggregate, which has been published upon in literature multiple times, especially at the lower pHs we're speaking of. So it's possible that the active hydrodynamic radius is larger in the insulin model than the lysosome model. And hopefully that will be probed when we go to the macro scale measurement of the drug release and SCISSOR experiments. The apparent diffusion coefficients based on temperature for the lysosome and insulin in solution and within the gelling network can be seen on the left, and a representation of the echo data can be seen on the right. And so on the left, it's shown for insulin and lysosome that the Stokes-Einstein equations both overestimate the amount of diffusion that will happen. In addition, both lysosome and insulin move slower within the gel than they do in solution. What this means is that it's most likely that these molecules are interacting with the gel structure in some capacity that is slowing their diffusion more than expected by Stokes-Einstein. And to know that this anomalous result is not coming from, or this unexpected result is not coming from some sort of mechanical interaction with the motion of the gel, what the graph on the right shows is that the signal that we're getting, that the authors are getting from that from the gel skeleton, is basically non-existent. So that most signal and deconvolution they're doing is coming from the APIs diffusing through that network. So to quickly review the SCISSOR results and the release curve studies, in the first row we can see the ibuprofen sodium solution injections as well as the gel injections. These images are immediately after the injectate was administered into the ECM cartridge, and gelling was allowed to occur over a 1-hour equilibration time. So with the ibuprofen, as suggested in the previous slide, or the previous image, there's some sort of aggregation that may happen that's causing the hydrodynamic radius of both ibuprofen and insulin to become greater than is expected. This can be seen with the ibuprofen solution sample, where you see this turbid response, as well as the insulin. It can be clearly seen in these images that even in solution form, these molecules are going through some sort of aggregation that may affect how they diffuse through these matrices. And then please note that along this, the insulin, which is a larger molecule, diffuses quite slower as compared to the ibuprofen, which is to be expected. But comparing the insulin and the lysosome molecule will give us more feedback or more information about how these biomolecules are interacting within this space as well. Comparing the lysosome and insulin release curves, you can see that there's quite a different action between the two of them, even though they are both protein-based molecules. The insulin, you can see, is a very slow uptake into solution, most likely caused again by that aggregation that is predicted by the authors within the literature and within previous literature. And when looking at the lysosome mechanism of action, there seems to be two phases of release, one being the immediate diffusion out of the gel as caused by individual lysosomes that are not aggregating to form a larger hydrodynamic radius, so they can probably move through the gel at a faster rate. This is backed up by the soluble nature shown by the lysosome in the solution injection, where a turbid response was not seen, So it's more likely that more ideally solubilized molecules are existing within this injection. And then one more change between the insulin and lysosome is that the lysosome, after an initial burst release, seems to then plateau and have a pretty stable rate of release thereafter, much more akin to a long-acting injectable. Thank you so much for sitting through all that data with me. I think the authors did an incredible job showing that microscale observations about diffusion coefficients and mechanisms of diffusion within gel and solution-based environments can be correlated with results that you get from a macroscale environment like the subcutaneous injection site simulator SCISSOR. And the pairing of those two methods can show you not only that your molecules are aggregating or will aggregate or are moving through a solution at a certain rate, but then you can actually see via bulk solution analytics that those properties are causing a difference in your gelation mechanisms, the characteristics of your gel, and the bulk diffuse properties of the APIs impregnated within that gel structure. If you have any questions about this study or others or possibilities in the application space for the SCISSOR, please email me or our product manager, Emmy. We'll be happy to answer any questions, and thank you and see you next time.